Okay, so we ended with coastwide objective. Would you like me to continue on then? Rest of the presentation? We're more than halfway through this one, so that's good. Okay, so another SRB request from SRB024 last September was about this maintaining spatial population structure, an objective. And it was to potentially define an objective related to spatial population structure based on unfished levels in those different regions. And we presented some of this last September and had not made any more progress since then, but I just wanted to remind where we're at. So currently, spatial spawning biomass is not one of those 4 priority objectives, but it is an important objective. And this objective was developed because, there was the thought that just conserving some spatial structure in the population may be useful, since we don't know what contributes to a successful recruitment in any given year. So what we had defined in the past are these gray bars on this plot on the right with the different regions. And we just sort of the Secretariat ad hoc defined percentages, proportion of the biomass that in each area that had been below what had been observed over the last 20 years. And so we just ad hoc defined some limits. And then from the MSE, what I've plotted in the colors are the proportion of the biomass, the range of the proportions seen in those simulations, and just to make sure they're above those minimum percentages. And that's why the SRB thought, well, maybe there's a better metric on this. One thing to note, in previous operating models, one difference between previous MSE simulations and these MSE simulations is this proportion in region 4B. It was the purple dot, which is pretty small confidence intervals on it, but it was a lot lower than that, and it often dipped into the below the threshold. So we could not actually meet this objective. But now that we've updated the operating model, region 4B, for whatever reason, has more biomass in it. But we're at the very tail end there anyway. So yeah, Sean?
Am I missing something here? Do those proportions sum to 1?
No.
Is it important?
The ones in the color.
Either
ones in the color should
the ones in the colors
pretty
probably
close. Yeah.
It's the other one. I'm not sure why that would.
No, because what we just defined that as a minimum, not necessarily. So
Their minimum.
What's that? No, the gray is just defining the threshold. It's not any observation or anything. So they're all above the minimum defined. The colors are from the MSE simulations. The gray bars indicate where we don't want to be from the simulations.
You could never be at all
Yeah.
values.
So we just want to define any one region independently. We don't want to go below that threshold. And what we see in the current simulation results is that we now meet this objective given the updated operating model.
What was the
I don't know exactly what it was. There could have been, yeah, I never really dug into that. But it was only for 4B, and I mean, we're talking a difference of like an increase of about 1 to 2% there. So it's, on the edge there. So
the exchange rates into
I think reconditioning the model changed distribution of recruitment and exchange rates. And then you had one of the models has the higher M on average. So maybe that had something to do with it too. So what we presented last time as well was in the operating model, it actually is running 2 populations, and one of them is unfished. So we can always do the comparison to the unfished spawning biomass. So what we've done here is actually calculated or presented the unfished or the proportion of the spawning biomass to unfished, so relative spawning biomass within each region.
I thought possible to make it a little.
Oh yeah, sorry. Can you maximize it?
Difficult to see.
Yeah, no, I get. Yeah, sorry about that. I'm just looking at my screen. Yeah. Thanks. And so these are the relative spawning biomass in each region, and just some the lines at 20, 30, and 36, which don't really have any meaning by a region, but I just put them in there as a reference. And what this shows is, I mean, there's a lot of uncertainty in the very start of the model, as you would expect, but you can see that region 2 has sustained some pretty low levels of compared to unfished levels, so some low relative spawning biomass over its time period. If you remember, the general movement of halibut is into region 2 as sort of a sink area, whereas regions 4 and 4B are source areas.
Alan, this reminds me, I think we've talked a little bit about whether a relative spawning biomass is the right criterion or an absolute, because you could go below the threshold, even if the population was increasing, if it was increasing more rapidly in another region. So, I mean, that's.
Or vice versa. You could be above the threshold when it's
declining.
at very low level, right?
Yeah, it's not declining. So
Potentially. Yeah, that's a good point. And to be honest, we had not made any more progress on this. And part of the reason is we're moving away from distribution, and we've been focusing on these priority objectives of the commission. But I do think it's good to make sure that we're bringing back in these spatial objectives for conserving population structure.
exactly. This is a very different spatial objective.
Mhm.
This is not about like equity or performance or economic considerations. This is about maintaining some biodiversity in the population that may be important under different environmental regimes.
Yeah, so to be honest, this just got put on the back burner this time as in progress. So that's what we have for that concept of spatial complexity objective. What I want to move into next are just sort of the management procedures we plan to investigate in 2024. And this is also stemming from a recommendation of the SRB as well as the MSAB. And this includes investigating assessment frequency with an empirical rule in the years without an assessment. Potentially putting constraints on the interannual change in the TCY to reduce the variability from year to year. We'll look at a range of fishing intensities, of course, when we do this. And then there's the FIST designs, which isn't investigating a management procedure to implement, but just more understanding the effects of FIST designs. Part of this, and I've included it in here, is a scientific discussion, just a technical discussion to have. And that's exactly, we still have to allocate in the simulations to the different sectors and areas. And so the question is, how do we characterize the uncertainty in the distribution? And we have a proposal on that.
Just clarify what's meant by assessment frequency there.
Conducting an assessment annually, as we're doing now, and presenting those results to the commission. Conducting it biannually or triennially, where in the years without an assessment, you would have an empirical rule that would provide the TCUI. And what past SRB recommendation was to look at it triennially, because that's our current process of a full assessment every 3 years.
I'm not sure exactly. I'm not sure that's what that means, at least in our recommendation, what we meant to say. So what's implied there, so the second line, frequency of the stock assessment elements of management procedures.
I think the
that
elements
implies that and if you interpret that as assessment frequency, the way you just said, which is fitting models to data, it's not what that meant.
I think the elements was actually referring to the management procedures in this sense, as the assessment frequency is one element of a management procedure.
So if it was some running average of the survey, this has to do with how often do you update the TCUI? Do you do every year? You do it every third year.
That is certainly part
That's
of
what
it.
that means.
Well, we've taken it a little further than that in that is one scenario that we have been investigating actually is doing an assessment and having a constant TCY for the next 3 years. But another element or another management procedure here would be conducting the assessment and then using the FIS results when they're in the second and third years to update the TCY up or down based on if the FIS went up 10%, increase the TCY by 10%. So that's been sort of what we've been investigating for the last 2 years or so.
So using a model.
Yes.
In the MSC, using a model.
Um,
Or that approximation.
Yeah, the MSC has always been using that approximation. The extension of all of this then is not even using an assessment to determine the TCY, but using a simpler model for that, and then using the assessment as a check-in every third year, or whatever it was, to determine stock status or whatnot, like the sablefish approach. And we haven't really gotten to that point, even though the SRB has been recommending to get to that point, using a simpler model to, as the management procedure to determine the TCY. The commission and the MSAB hasn't let go of the assessment as a tool yet.
This is what those things need to be done. Because, a whole bunch of work testing a stock assessment method that doesn't even exist.
Yeah.
It's approximating your stock assessment. Well, so does a running average of the survey.
Yeah.
And
Sure.
you know what I mean?
Mhm.
Whereas the running average of the survey, you could actually use as an MP.
Mhm.
So, this gets back to where we started, which is it would be nice if there was just some agreement that as of a certain date, we're going to adopt a procedure, which gives you okay, then we have to test procedures that actually exist or could exist,
Mhm.
and I think you would very quickly, it would be obvious to say this the most straightforward thing to do is a running average of the survey that do your assessment every second or third year as you're doing, and update the operating model every cycle and incorporate the exceptional circumstances check. Like all that stuff works together. So you're never far off.
Yep.
And all you're using the MP for is to set the TCY. And then whether you do it for 2-year chunks, 1 year, 2-year chunks, 3-year chunks is kind of the other thing.
Yeah. That makes sense. I think. Now you've been to the annual meeting and you've probably seen the stock assessment presentation with the decision table. There's so much presented at the annual meeting that is for their decision-making process, even at the coast-wide scale. I think that might be a reason it's been hard to move to a fixed management procedure is they want to see these tools to make a decision,
Yeah.
right?
Which, gets us back to I brought this up yesterday in a conversation about finding out what people's objectives are and what it is they want. And so that very thing is why don't they want to give up on the annual process? Well, why? Okay. The only thing I could come to is that people want something to talk about. They want something to do at the annual meeting because the annual meeting is important.
Mm-hmm.
They don't want to see if there's nothing to meet about, right? If there's nothing to negotiate over, then why go? And they're afraid of losing the annual meeting. Maybe that's the underlying objective.
They can still negotiate over.
Well, that was the point. There's lots of stuff to talk about
Mm-hmm.
that you could talk about otherwise.
It frees up time for you to.
Yeah.
Yeah.
But I don't doubt that you're right. Adopting a management procedure makes it's all superfluous, right?
Well,
Or it can be seen that way. It's like,
they have an important role in defining the overall objectives. We still talk about it. Negotiating the distribution of the TAC. Talk about that for a week. Whether going over other things, but there's lots to do.
Yeah, it will take time, I think, to move away from this paradigm of, because even during their negotiations of distribution of the TCUI, sometimes those negotiations include giving a little more to one area, raising the coastwide TCUI, part of the concept. So we've been, when I first came along and we were introducing that paradigm of coastwide TCUI for the health of the stock, conservation, making sure everything's good, and then distribute that. And so it's still in the concept of, we have this coastwide TCUI, let's distribute it and depart from that coastwide recommendation. MP.
Makes distributing it easier if you don't have to take from some other region to give.
It makes it a lot easier. Yeah. A lot of talk about zero-sum games, whether or not we're in one. I think, that's a really good point. And I think that the SRB has said similar things in the past, and it's something that we often present. So here is the assessment frequency and empirical rule, where we conduct a stock assessment for decision-making annually, biennially, or triennially. And in those interim years without a stock assessment, we would have some empirical rule operating in this way. And this might be a good stepping stone towards that end goal of adopting a formal management procedure. Baby steps, I guess. So we've been investigating 1 and 2 under the empirical rules, and then the dots, the bullet points, indicate some other empirical rules that we've been thinking about incorporating and haven't fully flushed out. But the first 2 are actually coded into the MSC simulation framework. The MSAB made a request at the last meeting, and that the Secretariat and SRB develop empirical rule options based on a static coastwide TCUI determined from the stock assessment, the FISO 32 WPUE, which are the first 2, but they also suggested we're incorporating commercial and FIS age data into this concept as well. And we haven't put a lot of thought into that, but it's something, that we are willing to think about. I'm not sure if I'll get implemented this year, but any advice that the SRB has on how we might incorporate age data into an empirical would be interesting.
I see that. I wonder if that's just kicking the can down the road, like not quite ready
Well,
to adopt something here, so we want
it
to keep requesting things.
Yeah, it could be. I think it also stems from the stock assessment presentation at the annual meeting. Made a lot of note that we're now relying on the 2012 year class quite a bit. We had this 6-year period of low recruitment that's gone through the fishery. The past year classes are dying out, and now we have one young year class that could become pretty dominant and relying on in the fishery. And there's a lot of uncertainty when that matures. You'll see maturity presentations later. And so that was a source of uncertainty that a lot of people are focusing in on. And I think that might be where this is coming from as well. But there's a lot of kicking the can down the road too.
Your operating model simulations reflect these occasional year classes, or what does the recruitment variability look like in the projections compared to?
That's something we haven't looked at. Yeah.
If you are,
Mm-hmm.
then your NSC is
Yeah, robust set. Yeah.
and it's going to be reflected in that SPR target that you're aiming for.
And the MSC does have the regimes, high and low, good and bad recruitment regimes. So it will have periods of low and periods of high, but those are more on the decadal.
I think I remember seeing, I think you showed some example recruitment series.
Mm-hmm.
And I remember looking at that, thinking, We've concluded it's pretty good.
Yeah.
Representing frequency of large year.
There was frequency of that in that, how often these regimes switched. And we adjusted that maybe a year or two ago. So it seemed a little off before that. But I haven't looked at sort of episodic improvements.
If there was some kind of metric you could derive, some year class dominance
Mhm.
metric. You could
Yeah, yeah.
projection.
And it's, and the literature's been coming up with more age-based metrics or metrics based on age structure in the population. Recently, the Griffiths paper that was recent, that's good. And I know there's some sablefish work going on in the US because they've had, reliance on really young fish. And so they're looking at metrics on what's the balance between young and old, I guess, fish in there as well. So
We're trying to incorporate sublegal fish in the management procedure so we can cap the discard.
yeah,
Because right now it's free for all because it's just a size limit. You can discard.
so that was part of this. Yeah. So, so we're thinking about some alternative empirical rules, but we'll definitely be doing these first two empirical rules in the simulations. One is just constant TCY, so the assessment and the decision sets it for a number of years. And the second one is it's adjusted by the FIS. And then I think it's important to remember there's decision-making variability in all of these simulations all the time. It's not always taking the coast-wide TCY from the MP, it's going above and below. And it's also distributing it with some variability as well. Related to FIS designs, I'm not going to spend a lot of time on this. You've seen Ian's presentation on this morning, what he's doing in the stock assessment. We plan to, with the MSE, just sort of take those results, similar design, and bring that right into the MSE. But that's something that's not likely to occur until earliest later this year. Once we get, we have results from the space-time model on all the simulations of these different designs, do the assessment stuff this summer, and then bring that into the MSE. So we're just awaiting on assessment results on that, but we'll incorporate a similar design. The distribution of the TCY, the operating model is 4 regions, but has 33 different fleets in it, multiple sectors within one region. And a lot of that was developed early on because we knew we were going to investigate distribution procedures. Now we're not. And so I questioned, do we need a spatial model? I think the spatial model and the multiple fleets has a lot of benefits for other reasons, for some, scenario examinations we have, just understanding. We've done some simulations I presented last year on effect of the environment and where biomass goes in different environmental regimes. So I think the operating model is useful in that sense. But what that means is we still need to distribute that across these regulatory areas, basically. And we just want to make sure we're doing that with a range of uncertainty that's reasonable, even though we don't know what the commission decisions will be on distribution, but we have past observations. So what we've done in the past with the operating model is we've used, I think it was 5 different distribution procedures that the commission defined as a potential range of ways they see they would distribute the TCY. And then we added some variability on top of that. It's called decision-making variability. We've been thinking more recently, why don't we just use past observations and what they've actually done and simulate from that? And so that's our proposal, and moving forward is to incorporate this new procedure. So for example, the TCY in regulatory area 2A, not shown in any of the plots, has been constant at 1.65 for the last 6 years. And it could be less if there was a conservation concern. The proportion of the TCY in 2B, which is Canada, has ranged between 17 and 21% in the last 12 years. And that's what's shown in these plots. We have the percentage of TCY and 2B over years. So you see in the early part of the time series, 2014, it was above 20%, but it's more stabilized just around 18.6%, I believe it was. And then we plotted the percentage of TCY and 2B against the total coastwide TCY, and there's not any general pattern there to be seen. It's been pretty random, you might say.
It's 1.65. What?
A million pounds. Sorry.
So it's an absolute.
Yeah, so the TCY and 2A has been an absolute amount of 1.65 million pounds. The proportion of the TCY and 2B has been these proportions. So the proposal here is distribute 1.65 million pounds to 2A unless there's a conservation concern and the manager, and we have to lower that for some reason. We'll figure out how to do that in the model. And then to simulate 2B from some distribution, and I've just plotted a triangle distribution over top of a smooth density distribution in gray on what these actual observations percentages look like. And you see it really captures well the more recent years, this triangle distribution. And I like a triangle distribution because they're easy for people to just specify what a min, max, and the mode is.
That captures that?
I think
What am I looking at? What's the smooth line?
the smooth line is the density over all of the observations here. And what I'm saying is it captures the more recent years where all these higher percentages were before 2018. So this bump out on the,
Sorry, I don't see what you're pointing at.
yeah, you don't. Let me get the pointer. There it is. So over here, these are all the earliest observations in the time series, where this peak is all the later observations in the time series. And so this is a proposal. We could just draw from these past observations as well in some way. We can do it multiple ways, but the thought now was just a real simple distribution.
Would you scrunch it between 17 and 19 and a half? That looks more like a triangle distribution.
Yeah, we could do that as well too. I just wanted to capture an occasional large percentages out in this region. That was my thinking.
Does it matter within this range? Take 17%, 20%.
Not in terms of the coastwide TCY. I mean, you don't think it mattered much, but it matters.
It won't change the fishing intensity at the bigger level.
Yeah. But when there's a lot of performance metrics that we do report at a regional and a regulatory area basis, which this would greatly matter. Now we can remove those performance metrics from the evaluation and not present those and just focus on the coastwide aspects as well. But on the coastwide sense, yeah, it doesn't matter if it's 17 or 21, or very small effects.
So then I guess the question is whether to remove them from the metrics that you reported.
The area or
Yeah.
area specific metrics. Yeah.
And maybe you just say, since the MSE is no longer looking at distribution, it doesn't make sense to report those because we don't have a consistent way to do it. It all, at the regional level, it all depends what you guys decide in terms of distributing the TCUY among the regions.
Yeah. Yeah. I mean, that's a good point. I just think when I go to the MSAB meeting, what might actually happen in reality, and that is we have different sectors and areas represented on the MSAB, and they're always interested in what's going to happen in their area. But I think maybe that's an argument to actually remove them and saying that's not what we want to focus on. The operating model is not really specified to be accurate at those levels, but it's specified to be accurate at regional levels.
Well, it's accurate at those levels if you're willing to have a management procedure that
Yeah.
distributes the quota, right?
Yeah.
But if you're not willing to do that,
Yeah.
then you've taken that piece with the highest leverage for the regional distribution and removed it. So
That will make the evaluation much easier too. Removing those performance metrics.
I mean, that is a pretty big change, though. Should we make sure we're all.
So what performance metric are we talking about?
Area
Or
specific.
TCUY?
Any area specific, like would be the TCY and 2B, you know, in the long term or the short term, the variability in the TCY and 2B.
So
So
that to me sounds like it should be included.
But it's hard to include if you're not willing to specify the distribution.
Well, like here, I assume what you're doing here is sampling from that, say, triangle. So you set 1.65 to 2A, sample something from the 17 to 20% at 2B, and then distribute the rest amongst other areas according to some other percentages.
which I'll show next.
So once that's done, you would have some output, and 2B could say, okay, our TCUY is, actually it's in this range.
Yeah, I wasn't saying it's difficult to implement. You can implement it, but it's difficult to know how well this will represent their decision making going forward, since that's constrained by the market procedure.
Yes, subject to behaving similarly as before.
Assumptions in this model.
Well, one option is adopting it.
I mean, one
Adopting it.
for sure. I mean, that's the best option A. But I mean, I wonder, since this is really designed to explore the space more, I wonder if you present 4 options, which is sort of each area at the high point of what it's received in the past, sort of sequentially.
So we were sort of in that boat when we had 5 different distribution procedures decided by the commissioners. So we had, you know, these areas low, these areas were high, and then the other end, you know. So we had this range of distribution procedures. And we were concerned about presenting each individual distribution procedure. I think a lot of people were concerned about that because that's one example. And I think there's a lot of concern about anchoring, anchoring on a specific distribution. The commission is in a negotiation process right now, and they're really concerned about results coming out that influence that negotiation in perhaps a way that is not complete. So we've been, I'm just being candid here. We're just being, just trying to represent that uncertainty. And so we've always integrated over any choices anybody's ever made. Just had it presented.
I feel like we've gone back and forth a lot.
Yeah, I think so. I think we've had this discussion in the past. Yeah.
I think going back, what I was asking about is if it was a matter of TCEY versus percentage of TCEY. If percentage of TCEY is a performance metric, which it could be if you were, say, using the old version by this. So that's, you're not using percentage of TCEY as a performance metric.
It's probably presented somewhere. It's probably available. We have this MSC Explorer tool with like 200 metrics, and I'm pretty sure it would be a
That's the one that I would kind of want to de-emphasize because that,
yeah. If we did present the TCY by area, I'm sure people would calculate the percentage based on those outcomes, which would not be accurate in that sense. You got to do it on a simulation basis. So that's my worry. They would ask for it.
yeah, I mean, it's hard to,
Yeah,
it's
exactly.
hard for 2B to say, oh yeah, great, you know, Halibut coastwide's doing great.
It is.
Matters to them. And how it does coastwide does matter to them. It does affect them.
So that quota matters to every single area.
And if you're including the uncertainty in how the decisions are going to be done, as well as in the the operating model isn't doing great
No.
on distributing fish.
No.
So there's that, even if you did have an MP apparently controlling the actual distribution, it still wouldn't be that reliable because the operating model isn't
Yeah.
that great in spatial dynamics.
Mhm. So, yeah, but in the operating model, we have to distribute it some way. And I think the reporting of performance metrics by area is a separate question from this. If we didn't want to distribute the TCY in the operating model, we'd have to go back and build a coastwide operating model, single area.
Yeah.
We could do. We did in the past.
Dumbing down the operating model seems like.
So the proposal here is to use past observations. You know, we don't have to use a triangle distribution like this. We could sample from past observations or make this a beta or do something different, you know. But I'll show you what we were thinking about for the Alaskan areas. And that was to first just determine the TCY in 2A and 2B using the method on the previous slide. And then we're left with an Alaskan TCY, and then we distribute that in some way. And we would simply just use a multinomial distribution to do that, tuned to the past observations, which is exactly what this plot shows. The blue dots are the observed percentages in each area. And so you can see there's an outlier there in 2C, and it seems like it was taken up by 3A in that. This is a early year. I can't remember, maybe 2013. Oh, sorry. This one, I always forget. So yeah, there's that outlier there. But just tuning this multinomial distribution with error, you see you can capture those past observations pretty well and have the correlation between them.
What about just doing random draws of years past, so you preserve the sum to one?
Yeah, you could do the years.
And
So-
maybe sample more frequently from the more recent years.
That's
So that's representative of what's happening now a bit more, but also incorporates the past.
Yeah, that's a really good concept. I'm thinking about how to incorporate 2A where decisions on 2A have not been based on a percentage in recent years. They've been based on the flat 1.65 million pounds.
You could still just
Do that.
select
and
a
then-
year, take
yeah.
the absolute value from 2A and then the percentages from the others.
You'd have to renormalize those percentages because the overall TCY would be different in the simulation. Yeah.
Do you even need to do it at all? Like, it's just so tight that if you ignored it, if you ignored the random variation and just use the fixed percentages, how far off are your performance metrics going to be?
But you say that this matters a lot for people, right? Whether they're the top end of the gray bar or the bottom end of the gray bar.
When we start talking about this, it becomes pretty in-depth discussions. And I think from my experience, I see people look at two things, and those two things are how much TCY do I get in my area? Well, three things. How much TCY do I get in my area? How much does that change from year to year in my area? And what percentage do I get in my area? So they are aware of that in what I've experienced in my in here. But yeah, I mean, I'm open to easy, simple things. And like, as you said, this range, it would be actually an interesting experiment to do some simulations with just one fixed range and then incorporate something like this and see did it make a huge difference given all the other uncertainties in the model.
The bootstrapping of years, I think it's easy to explain.
Yeah.
You don't just, pick a year at random, use the distributions that you have for that year. So it's to get the overall distribution certainly.
I do like that idea. It's more simple than this.
Over the longer historical period, though, the distribution has changed pretty dramatically in terms of the catch. There were time periods when the majority of the catch came out of region 3, and other time periods when the majority of the catch came out of region 2. There's been some huge swings. I just don't know. This is all pretty localized to recent stock conditions. I'm not sure. If you could develop a pretty big mismatch in with the operating model if you had a very different true stock distribution compared to what you're applying. I don't know.
That's
you
true.
Bootstrap from recent years where you think that distribution is the spatial distribution of the stock has been still representative.
Hear totally what you said, but if you had a baseline simulation that distributed TCY directly in proportion to biomass, it's a reference case. I don't know if it would get that extreme now. You know, to go
Right.
back to levels like you in 2B that you had
Right.
or wherever, you know, I don't know if that's even possible anymore, but
How different is the distribution of TCE?
Well, when we would present to the commissioners a sort of default or reference distribution, it was based on the distribution of the biomass observed in the FIS, adjusted to put a little bit more TCY in region 2 and 3A. Just because they thought they could withstand higher fishing intensities than the western areas. So it has always been shifted typically away from western areas into the southeastern areas. But yeah, so it differs slightly, but wouldn't say it's far off. It's always, you know, even after the commission decisions, they are always considering how the stock is distributed. And we did talk with them at the last meeting about they still have some interest in how the stock is distributed regionally. Not maybe to the regulatory area, but how it's distributed regionally.
yeah.
Yeah.
That
So,
would track,
mhm.
because part of this is that you bias and movement towards area 2, so parts of area 3. Fishing mortality can be higher. Yeah, proportional to biomass.
Yep.
Because of that. So,
Should I add uncertainty on that at all, or just have it fixed with that? Just as a,
just as a reference to say,
yeah.
and then look at how the distribution compares to the, at least theoretical,
Oh, okay.
biological.
Yeah. So we, when I showed that illustration of the harvest policy in the very first slide, and then the distribution got wiped out. One concept we were thinking of is focusing on the coastwide as a management procedure, the TCY, and then actually having a reference distribution procedure to inform the negotiation process at the annual meeting. But that didn't go over very well. That was an anchor, right? That's an anchor point. So whatever distribution procedure I use in the model, I do have to be careful about. And I really want it as hidden as possible to not refocus people onto that aspect of the evaluation, to keep them focused on the coastline evaluation. And that was sort of why we thought, well, let's just throw in a bunch of uncertainty so nobody knows what it is. But maybe that's not the best route forward.
I would just go the other way and just not have uncertainty there and say the uncertainty in the distribution procedure is entirely in your control. If you want there to be zero uncertainty in that, set a rule and that's what it will be.
It's actually a good point.
But if you have negotiations that really change it from one year to the next, that's added uncertainty that you're putting into the process.
Mhm. Okay, well, I may move on from that in the interest of time. I think those are great ideas. It sounds like, well, I do want to have something sort of buttoned up
Yeah, so let's get a clear recommendation.
so that I can do simulations this summer. Yeah.
We're suggesting keep the distribution component of the policy black box. But in the operating model, you want or not?
In the operating model, distribute proportional to
Well, no, I was saying as a reference to see what the ideal distribution of biomass would have been.
Mhm.
Because I think the question, getting back to what Ian was saying, is that at times, the distribution has been very disproportionate. Distribution catch has been very disproportionate.
Well, it might not have been so far from biomass, but the biomass itself has changed dramatically. That's why I was worried about having fixed percentages and then the biomass changing in the operating model. But if it's, I don't, well, it's also been biomass. Both of those things are true. There have been periods of years when it was very far from, I mean, for 2 decades in the early 1900s, it all came out of region 2.
Yeah.
So it's way out of proportion to the biomass.
Probably isn't going to happen again.
Probably not going to happen again. Yeah.
So something like this, I think what we just presented, 1.65 triangle distribution, and this is a pretty decent reference. In the
And then compare that to proportional to biomass
Well-
and just see how far off.
I don't want to add new work, or I'm not sure that that's going to help anything, because they really didn't ever like that idea. The whole apportionment thing,
Yeah.
that's kind of raising that issue, and I don't want to do that again. But I think we're talking about either use deterministic simulations from these, or random. But you got to do random for 2B because it's quite a bit variable.
Maybe there's a question for the MSAT. Yeah, we need to do something.
Yeah, we talked to them about it. Yeah, I think the what tends to be there is people get so focused on their area that it becomes a discussion about what is the best way to do this, and then they want to evaluate it. So it's really a technical question for the SRB, this aspect of it.
So we have some options. So one is distribute according to 1.65, draw from the triangle, and then these use the fixed percentages. Just use the mean of each one. That's one option.
Yep.
Another, which I liked, was when each region came up with their own scheme, and you just sampled from those schemes.
What do you mean by ski?
Isn't that what you were doing before? There were like 5 different
It was
distribution
the commissioners
options.
came with 5 separate distribution options.
Yeah, you
right?
simulated all of them, but didn't tell anybody what was what. You
Yep, just
pooled it,
integrated.
which I think is a great idea.
I could still
to,
do
to
that.
let them do it, right? Their input is now involved, but it's in proportion.
Yeah.
Its impact is in proportion to the importance of it and their size, I guess,
I mean, that's already
in a
coded.
blind way.
I could easily do that.
So that's an option. Maybe we can discuss that more to see.
Yeah. Third option, another option was Olaf's suggestion, just sampling from past observations and maybe with a higher frequency on recent.
Yeah, so the 2B is you're sampling from the past. Here you sample from the past, which is probably the least controversial of all. It's simplest.
Yeah, definitely.
So
One benefit of this, one benefit of the approach that I've presented here in doing 2A and 2B first and then focusing on Alaska, the conference board last year was very interested in using that as an approach. And, there seems to be a lot of negotiation about distribution to the 2 parties, the 2 countries. But Alaska is somewhat interested in developing a management procedure for distribution among Alaskan areas. And they have a little working group outside of the MSE process that's been investigating, or talking about it at least. And so this would incorporate a easy way to bring their ideas into that if the commission was to go down that road. But I don't see the commission going down for a while.
okay, so I would say, like, for now, for this iteration of your simulations, we're going to recommend, as terms of a distribution for the simulations, set 2A 1.65, set 2B from the triangle distribution, set Alaska to the historical distribution.
And just a fixed or sampled from historical.
Sampled from the historical distribution.
Okay.
And then you have to renormalize.
Well, Yeah, and that'd be easy. It would be just sample from the already renormalized Alaskan distribution. So, but when you sample a year, then specific year.
Then, according to our other recommendation, that you separate the TCY and then annual negotiations, have the flexibility to do what you want every year, that still holds, right? They still do it. This will account for that to the extent. It's probably.
They're not way far away from that distribution. It's going to be close enough.
Right, and it's part of maybe not exceptional circumstances, but something you're going to track over time is the distribution,
Of the
actual
TCY.
distribution compared to this one that you've been seeing.
That actually makes sense. That brings in, that's getting closer to bringing in fishery dependent data into the exceptional circumstances as well.
It's also consistent with what I do when I create the decision table every year. The decision table depends on the distribution as well, and I don't know it when I create it, but I just approximate what's been done recently. They've never deviated far enough to change anything beyond the rounding in the table. So
Yeah, if it doesn't affect the assessable,
it's the same logic.
project out longer, you're still getting.
So you're pretty well. Too.
No, but I haven't been far enough off for it to matter.
That's a pretty good prediction. Okay, so the last topic here is depensation and a request. I don't think I put the actual request, or maybe it's on the next slide, but it was a request to do a stress test in the MSE on depensation. So I just went back and reviewed all the papers about depensation and found a lot by you, Anna. It's really good work. Really cool stuff. And what I just wanted to remind everybody about depensation is when the per capita growth rate decreases when the abundance is low. So it doesn't, it's like has a harder time of reproduction success at low biomass. And what I've been using to begin to implement this and examine this is this formulation of the Beverton-Holt. And I like this formulation because all these parameters have some sort of meaning, where alpha is the asymptote and beta is the spawning biomass that produces alpha over 2 recruit. So I can get a really good idea what this looks like. And then the delta is the depensation parameter. And then if we look down on the right plots, recruits per spawner, plotted against spawners, we would expect to see these shapes. And with depensation, you see this increase and then the decrease. And so I just wanted to remind people, because what I did is, I'll show you in a moment, I actually looked at some data or some model results of that. Before we get into that, we just had a little roundtable here within the office about, well, what are some mechanisms in Pacific halibut for depensation? What would why would halibut experience depensation? And, any spawning biomass gets to 0, of course, they have no chance of reproductive success, but at low spawning biomasses. And so we thought about environmental effects, which is a lot of work Anna's been doing. And we thought, well, halibut are definitely experiencing environmental effects. They've seen this regimes, the good and bad PDOs, and we thought there's definitely possible depensation, say in a low regime and maybe not in a high regime. So we want to be aware of that. Predator-prey interactions, increases in natural mortality. We thought probably not likely, given Pacific halibut are a generalist, have a wide range. They are often the top predator. They may have some similar predators in competition, so maybe there's a chance of that depensation if a predator is out competing them in some way when halibut population is low. So something maybe not likely, but possible.
The top predator when they're adults. But
true.
through life stages where
Yeah.
could it be the cultivation, depensation, trophic triangle thing going on? As is, has been proposed for cod and
Right,
forage
right.
fish, right? Where the forage fish are eating the cod eggs and
Yeah.
they're crossing.
Did Grant sort of start bringing that into his multi-species models, or I thought I saw that somewhere recently, but
Well,
maybe it wasn't
It
how.
applies for some species, but
Similar enough for halibut.
They grow really fast when they're young, and yeah, I don't know, but definitely, and that's the kind of things we're thinking about. Like, what are some of these mechanisms? What should we be looking at? I forgot to include that one in here. Reduced probability of fertilization, maybe not likely because halibut tend to migrate back to spawning areas, so they might be attracted to form aggregation. So there is not really a reduced probability of fertilization. If there's two fish left, maybe they'll find each other.
on that one, if they do tend to congregate fairly strongly to spawning areas, that would make them susceptible to disease outbreaks and things.
Oh yeah, true.
Might have
Attract predators.
and predators, but
Yeah.
if thinking of parasites and diseases as kind of predators. I don't think a
we
big
don't have any
spawning
idea what casual
Tasman.
mortality on large halibut
Yeah.
is.
That's what they-
What is the leading cause of death of large halibut? I don't think we have any idea.
Probably fishing.
Yeah,
Well,
it's
yeah,
recreational
after-
fishing. And then group dynamics similarly in that they're feeding and spawning migrations. There's not really, when they do need a group, they tend to congregate into a group, and the benefits of being in a group may not be as necessary.
But this is depensation. A lot of this stuff is describing depensation mostly of adult health.
Yeah, true.
And you're talking about depensation in a spawn recruit relationship. So I think anything that causes depensatory mortality could apply to recruit.
Yeah, true. You're right. I mean, my focus when I brought this to the group was thinking about adult health, but thinking about other components.
It's a good bycatch. Could be.
Mm-hmm.
If the distribution of recruits is spatially like a basin kind of thing, and fishing right through the middle of the basin to the,
Yeah.
to the best part of the basin, that would generate depensatory fishing mortality.
Which could be the case.
These are kind of complementary effects.
Mm.
And then the emerging demographic effect is just a pattern that emerges, but it doesn't take any very standard saying what is the mechanism causing it, but it's more like emerging problem.
Right. A depensation emerges from any combination of these potentially. So what we looked at first before diving into the simulation is using the two long time series stock assessment models. We took the spawning biomass estimates, and you can see they're different between the two long models, areas as fleets and coastwide. And then we plotted the recruits per spawner against spawners for each environmental regime. And you can see the environmental regime across years by the different colors in the plot on the top plots right here, the blues and the reds. And so in the bottom right plot, we see sort of the classic shape of recruits per spawners without depensation. Here, maybe you argue right there, there's a little bit of a curve over. Hard to say. These are estimates from a model output, but
I would say that in the right hand, there is actually a little bit like decrease in the, like if there was depensation, then the curve would go more steep and
oh, I
steep up.
see.
But at least the red dots, the little triangles, they start to seize a little bit.
Yeah, they really.
Just the thing that what happens in that area where there is no data.
Right, and that's sort of
hopefully never see again.
yeah. But I mean, that's the issue, is that we haven't seen really low spawners to really see any sort of potential effect at this range. But all of this, let me go ahead. So I think this is a good investigation, but it certainly, I'm not arguing not to do the simulations, to do the stress test.
Well, I mean, this is exactly why we do MSC kind of stuff, is you say, okay, well, we have no information. That's all you know.
Yep.
You've got a trend that's going up, but you've got zero information on what happens after. So you can extrapolate the trend going up. You can extrapolate it going down. There you've got two scenarios. From both. And you kind of know what's going to happen in the ascending one, but
Right. So that's our proposal, is to do exactly that, is to do at least 6 simulations for a depensation stress test. And that is no depensation and with depensation. And to do each one of those at 3 different fishing intensities. One is a really high level that's actually at the fishing intensity we believe is analogous to MSY, but way above our reference. And we've experienced fishing intensities that high in the past in retrospective. But the question though is really, you know, how much on these levels. And what we may do is just define a first level of depensation and then start examining alternative levels of fishing intensities and depensation to see where the real effects start to come in. Like, are we talking the smallest amount of depensation has a big effect on management outcomes? Or are these fishing intensities, you know, the interaction between fishing intensity and depensation? So it's going to be a little bit more than 6 simulations because we don't know how much depensation to put in and what fishing intensities are going to result in negative or reduced outcomes, management outcomes.
I would maybe start by fitting the stock recruitment model and simply looking what kind of value it produces for this shape parameter. That
Okay.
would be much easier than to
Yeah.
any simulations to start with.
Yeah.
I agree. I'd tie it to the data so that you're not testing unrealistic scenarios. But instead, you're testing the whole range of possible scenarios in that left-hand side there where it's not tied to data.
But it's surprising that sometimes the earliest signs of depensation can actually come at the something like 60% of the carrying capacity. So well in the range where the data, there is data. This happened, for instance, for 3PS Atlantic Chord. We identified the inclination point where the curve starts to shape its form. And that was something like 60.
But that seems not plausible for halibut, right?
I don't know. It would be good to just fit the model and test. And
Yeah, I
see.
think because it's a third parameter, it's going to affect the whole thing. It's going to affect, like, it may not be visually distinguishable just looking at the data, but if you put the models in there, they might.
Yeah, that's good. Yeah, so we'll start with that, but you know, I'm not sure what the estimate of depensation will be from these data. And then I still might increase it just as a stress test to see what happens.
Yeah.
The what ifs, like how high does this need to be before we really start seeing effects?
Maybe fit it with a couple of fixed values of that delta parameter and then estimate the others, right? So that it is tied to your data, but also exploring alternative scenarios.
Yeah, and we can almost do like a likelihood profile over a delta or something like that.
And of course, depensation can change in time.
Yeah. So that was another thing is, should we be tying that to these different environmental regimes potentially? It looks like the recruitment spawner recruit relationship is different. So I mean, average recruitment changes with the regime, and because we parameterize it with steepness, that does change the stock recruitment relationship. something I've been trying to wrap my head around in the last couple of years. Okay, so that's the proposal there. I think that's really good advice, just trying to tie it back to the data. And those are, yeah, so I think that's the end.
And so it's not depensation, yes, it's depensation, and then each row is a different delta.
Well, it would be the same delta for 3 fishing intensities, and then maybe another set of rows for a different delta level.
Delta of 1, being a depensation, delta.
Yeah.
of
0.8, and then maybe a delta 0.6, but do it for at least 3 fishing intensities, low, reference, and high, to get an idea.
times the 2 stock recruit relationships.
Yeah, well, the stock recruitment relationships are tied into the one simulation because it's cycling between the two,
I got you.
unless you think it's important to bring those out and see them as individual, which I've done in the past.
So this is, the whole simulation involves changing the operating model to
Depensation.
depensation. And then each of those 3 fishing intensities.
Yeah. Yeah, but it's easy to have it always a low regime or always a high regime. That's just an input to the model. So I could separate those out if desired as well.
Well, your current model samples the PDO.
It simulates it cyclically.
Yeah, so I would just stick with that. Well, each machine.
It's more representative because it's really decade-old oscillations. And I think tying in 10, 20 years of low with 10, 20 years of high, and then those sort of correlation, that's useful. Yeah, so that's the end of my presentation. And here's just potential recommendations, which are just summary of everything that I've presented. And I really appreciate all the insight. That was really good discussions that we had today. Really appreciate that.
We've been including elite effects in our hearing models now
Oh, yeah?
in the form of, well, like, we did a similar thing where we estimate natural mortality over time. But when you look at that versus the biomass, it's very strong.
Yeah.
And so we put a model in there. So now our base operating model is density for natural mortality.
Oh, wow.
And because it's a function of biomass, it also depends on the body weight. So changes in weighted age start affecting things. It starts changing the natural mortality rate. So you can get a change in body weight, flips the thing on a downward spiral. And the dynamics change a lot in your sustainable harvest rate goes way down.
And of course, in the case of halibut, we do have the decrease in weight age.
Yeah, so that for this, I noticed that you were looking at spawners per recruit. Recruits per spawner, is that per spawning biomass or per spawner?
That would be spawning biomass. Yeah, I think
It's
per
per
spawn.
biomass. Then, yeah, that's, so changes in weighted age alone.
Yep.
Will change your spawning biomass, which will change your. In So a change in weighted age would have changed it anyway.
Yep.
And now it's going to have a double effect.
Mhm.
The other weird thing that happened did us when we went to do we started calculating reference points. So our first set of reference points, so you can't solve it analytically. Maybe you can here, but but we couldn't. So we simulated.
Mm-hmm.
So we simulated it with deterministic. So you do the deterministic simulations. And then you do just do some stochastic simulations to make sure it matches. Sure enough, it doesn't.
Yeah.
Because process error,
Feedback loops.
now the variance of the process error
Yeah.
actually affects
That's right.
your sustainable harvest rate.
Yeah.
Boy, you bring this up in a review meeting and just everybody's head explodes. You can't understand anything. Because now you not only have to simulate reference points, they have the full stochastic ones. Yeah, it really. That's your, it's a tough one.
Separation of science and management working.
Well, the manager's freaked out because the unfished biomass changed.
Yeah.
You're just getting used to it.
All right. Well, thank you very much. Anything else? I'm open to discussion. And if
Thank
you
you.
have any comments on anything, happy, even like the formulation for depensation or the equation, if you have alternatives there. I know you've worked with different ones.
Yeah, the sigmoidal Beretta vault is quite common, but there is also this extension that bases on rigor
Right.
silo load.
Yeah. Yeah.
Thank you.
There's also compensatory activity function.
Right. Yeah. Yeah. That was, you were on that paper, weren't you? Yeah. I looked at that. I just grew up with the Learman Hillborn,
But that incorporates other potential mechanisms
That's true.
besides just in the stock recruitment relationship, right? So it might be that there's density growth.
Yeah. What I've been thinking about too is, how to bring that into the operating model itself. And this would be pretty simple to do.
Maybe let's take a minute break. We get through biology. And we have till 4 o'clock for that.
We planned on that extending past this block, so we're still up ahead of schedule. So we have a whole empty block tomorrow afternoon.
And we already did AI, which was supposed to be tomorrow, right?
We're comfortably ahead of schedule still.
Thanks for that, Nacho.
You can enjoy your steak comfortably tonight.
I was already planning my night, studying my presentation.
If you hadn't told us, I don't think any of us would have noticed.
No.
Now I just think that it will be even better.
Camera and the mic for 10 minutes.
Yeah.
Water the coffee machine.
All right. Well, we're gonna start with a report on current and future biological and system science research activities. All right. So the activities that were conducted on biological and system science research are actually contemplated within our current 5-year program of integrated research and monitoring that defines research activities in 5 main areas. And those are the ones listed here: migration and population dynamics, secondly, reproduction, third, growth, fourth, mortality and survival assessment, and fifth, fishing technology. And all these research areas have actually been selected, and the particular topics have been selected because of their relevance in relation to the research priorities for both stock assessment and management strategy evaluation. In this first table that I'm not going to go into detail, you can see that in green and purple, those are the research activities in the research areas of reproduction and migration and population dynamics. Those are the top priority for stock assessment. And then for MSC, which is this next slide, both migration, population dynamics, and reproduction are some of the top research priorities also for MSc. And today what you will hear is primarily major updates on these two research areas. We're going to start with migration and population dynamics, and we're going to actually focus and provide probably the first updates on population structure. So we're going to be covering the area that is highlighted here in red, and I'll let Andy Jasonowicz speak to that. So if you want to move the cursor through share the computer.
Okay, thanks, Trisha. Yeah, so the next set of slides here is an update on the progress that we've made on our population genomics project. And for today, I thought it'd be nice to just start with a brief background and introduction, just to reorient everybody to the study design. And then from there, move into results that pertain to the analyses of population structure that we carried out. So we'll start with some principal components analysis and k-means clustering. This has been presented at meetings before, and then from there, moving into additional analyses that have been carried out since the previous SRB. Okay, so the primary objective for this project is really to resolve the genetic structure of Pacific halibut in IPHC convention waters using modern genomic techniques. And so to address this, the samples that we have available to address this are shown in the map here. And they're collected from 5 different geographic areas: British Columbia, Central Gulf of Alaska, the Bering Sea, the Central Aleutian Islands, and the Western Aleutian Islands. And these samples were collected over a fairly broad time frame between the years of 1999 and 2020. But kind of the key, most important aspect of these samples is that they were collected during the wintertime, which is the spawning season for Pacific halibut. So this is, the most accurate representation we have of spawning groups in these areas. Okay. So the method that we're utilizing to generate the raw data for this project is called low coverage whole genome resequencing. And this method allows for screening genomic variation at very high resolution. Essentially, this method has the capability to sample every single base pair of an individual's genome. Like I just mentioned, the primary application of this data is to establish a baseline of genetic diversity for spawning groups of Pacific halibut within IPHC convention waters. And being that this is a genomic approach, we can also use this data set to identify potential local and or environmental adaptations. So the data set that we've generated to date consists of, after our initial kind of quality control filtering, consists of 570 individuals, and this data was generated across 3 sequencing runs, and we're observing an average genomic coverage of 3.5x. And a simple way to think about this is on average we're sequencing an individual's genome 3.5 times over. The initial bioinformatic processing and analysis of this data set has enabled us to identify just shy of 10.4 million SNPs and autosomal regions of the genome. And about 4.8 million of those SNPs have a minor allele frequency of at least 0.05. So this is a common filtering threshold that's applied for, or that's used for some of the analyses that we have planned. And so while this indicates we have a lot of very low frequency mutations in our data set, there's still plenty for the analyses that we have planned. Oh, nice. Okay, so to start, one of the very first things we did when we generated this data set was conducted principal components analysis and PCA. So PCA is a dimensionality reduction technique. So it's a very nice tool for visualizing these high dimensional, very large genomic data sets. And again, this is on the base. This PCA is based off of those 4.8 million autosomal SNPs with an allele frequency of 0.05. And individuals. So in this figure here, these are individuals plotted along the top two principal components. And what you might notice is that, so I guess, yeah, so sorry, back up here. So each point represents an individual, and each individual is colored by the geographic area in which they were sampled from. And those lines represent 95% confidence ellipses associated with each, sampling area. And so you'll notice that there's a lot of overlap among the areas that were sampled for this study. And we're not able to actually partition the data set into any sort of discrete groupings here. You'll also notice that we're capturing very, very small amount of the total variation in the data set along these two principal components, indicating just, a general lack of structure in the data set. And so I guess to kind of go beyond just the method of the eyeball, we also conducted k-means clustering utilizing the top three principal components. And so these two figures here show the total within cluster sum of squares and BIC for the various k values we tested. That's between k of 1 and k of 20. And again, just examining these sorts of metrics, we can't make a very confident determination on saying that there's a discrete number of genetic clusters in our data set here. It's most likely that there's just kind of a single panmictic group based off of this. Okay, so moving on to I guess kind of the new analyses that we've done since the last meeting. So at SRB024, the SRB requested that admixture analyses and assignment testing be conducted and reported at SRB024. That is this meeting, including estimates of assignment accuracy. And I guess before we get into results, I would just kind of want to just introduce these methods and sort of what's going on with these. So, admixture analysis, these I guess would fall into a category of being unsupervised model-based clustering methods. And I guess when I say model-based, what I'm referring to is the fact that these models were developed specifically for genetic data, so they make specific assumptions about the genetic aspects of the data. But essentially, what's going on here is these are partitioning the data set into a predefined number of discrete populations, also referred to as K here. They're also estimating allele frequencies for each of those clusters. And then what we're most interested in the context of this request is these models also estimate membership probabilities for each individual to each cluster. Assignment testing, on the other hand, this is a supervised method, meaning that it requires us to predefine our population, the populations that we're attempting to assign our individuals to. And kind of the core question that these methods operate on is really, what is the likelihood that an individual's genotype originates from a set of predefined populations? Okay, so for the admixture analyses that we conducted, we used a piece of software called NGS Admix. This software is designed specifically to accommodate low coverage data. And we ran the model for a range of K values between K of 2 and K of 8. And we ran 5 replicates for each K value. As far as assigning individuals to a cluster, we required that an individual have at least an assignment probability of 0.8 to actually assign that individual to a single cluster. If that criteria was not met, those individuals were categorized as unassigned or admixed. And then in order to evaluate kind of support for the best value of K that we tested, we used this delta K statistic. And this statistic was developed specifically for these types of models and has been shown to be able to reliably detect kind of the highest level of hierarchical structure in your data set. So we have the delta K values associated with, in this figure here, the delta K values associated with each K value tested. And you'll notice that the highest value of delta K is associated with K of 2. So that might lead you to infer that we are dealing with 2 clusters here. However, there is kind of a key limitation of this statistic is that it cannot actually be used to actually detect the number of clusters when you're dealing with a K of 1 or sort of like a panmictic scenario. And with the next figure, I'm going to explain why I think that's what we're dealing with here. So these are confusion matrices for each value of K tested. So we have each panel is a different K value. So you have K of 2 on the left and K of 8 on the right. And then on the x-axis, we have the assigned clusters. And then on the y-axis are the geographic area, the sampling areas. And then in each cell is the number of individuals that were assigned to a specific cluster or categorized as unassigned. And I'll just say, what to take away from this here is in all cases, we're not able to confidently assign a majority of the samples to a single cluster. So most of the individuals in our data set are being categorized as unassigned. And while, even a K of 2, we're able to assign some individuals, that drops off very quickly as you begin to partition the data set into more groups. And what I think's happening here is essentially like, as you do that, you're less likely to observe that assignment probability of 0.8 for an individual by chance, as you partition the data set into more discrete clusters.
And if you look at any other values than 0.8.
No, just kind of, it's an arbitrary threshold. And I kind of drew from the literature, like I've seen, situations where you see as low as 0.7. And then in some applications, they're even as high as say like 0.95. So I felt this was kind of a reasonable, reasonable place to start. But I think
It's not going to change the picture.
no, it's not going to change the picture. Yeah,
It's
exactly.
not gonna look great.
Yeah, and if you actually, look at the distributions of kind of the highest assignment probability for individuals, you could go even lower than 0.8. It would again, it's not going to change the story right now. Okay, so moving on to assignment testing. So while the unsupervised clustering analyses we performed failed to essentially detect any sort of structure in our data set, we still can attempt to assign individuals back to the geographic location in which they were sampled using a set of highly differentiated SNPs. And so in this case, the software that we use to do this is WGS Assign. Again, it was developed specifically for low coverage data. And basically, so instead of, we essentially assign our reference populations were defined by the geographic area in which these samples were collected. And then to evaluate assignment accuracies, we followed a simple training holdout procedure. And this is the kind of the recommended approach when you're selecting SNPs based on allele frequency, which FST is sort of a measure of variance in allele frequency. And this may be a conservative approach, but it is kind of the recommended approach, and it's referred to the gold standard as a gold standard approach by a couple of prominent authors here. So, basically what we did here is we split the data into two portions, a training set and a validation set. And with the training set, we first estimated pairwise FST among the various geographic areas that we sampled. We pooled sample collection. We pooled across sample years. So we had basically 5 different areas. So that equated to 10 different pairwise comparisons that we're making here. We ordered those SNPs based on FST and selected the top 1,000 SNPs per comparison. We also required those SNPs to be at least 10,000 base pairs apart from one another to minimize any effects of SNPs that might be linked to each other on a chromosome. And then after we pooled all those lists from the pairwise comparisons, we removed any duplicate SNPs. So that left us with a SNP panel of 8,497 SNPs, if you will. And then from there, we proceeded to define the reference populations or reporting groups by estimating allele frequencies using only the samples in the training set. From there, we used the validation set. Essentially just, those individual, we assign those individuals in the validation set back to the reference established using the training set. And from there, we can obtain an estimate of assignment accuracy because the model hasn't seen those samples yet. So the assignment rates that we observed was the assignment rate that we observed was 34.7%. However, let's see. So however, all of those samples, all of the samples in the validation set assigned back to the Gulf of Alaska. And you can see that in the confusion matrix here. Again, we have the population of origin on the y-axis and the population that these individuals were assigned to on the x-axis. And they're all assigned back to the Gulf of Alaska with very, very high probability. Like these all had assignment probabilities that all of the samples had assignment probabilities that exceeded 95%. So to kind of take a look at our training set, we looked at the self-assignment rates of the training set using leave one out cross-validation. So if you're not familiar, you basically remove one sample from your data set or from the training set, re-estimate the baseline allele frequencies and see how that individual assigns back to those reference populations. And we observed 100% assignment accuracy within the training set. And what I think is going on here is that we're essentially capturing noise in the data set, noise associated with the samples selected for the training set. And since we have the largest number of samples collected from the Gulf of Alaska, we have the largest number of samples, and therefore we're obtaining the most accurate allele frequencies. If you assume, for the stock as a whole, if you assume there's just kind of a single panmictic group, those allele frequencies are going to be most representative from the stock as a whole, and therefore it's individuals are being I don't want to say by default assigned back to the Gulf of Alaska, but they're capturing all those individuals. So in conclusion, the unsupervised clustering methods that we've applied to this data set have failed to identify any sort of discrete genetic groups of Pacific halibut within IPHC convention waters. And we're also, this also limits our ability to actually assign individuals back to the location which they're sampled, which is not all that surprising. And it's highly likely that there's a considerable degree of gene flow that's occurring over very large spatial scales here. That's leading to the lack of structure that we're observing. And given our knowledge on Pacific halibut movements, this is should not come as such a surprise. We know that they move a lot across all life stages. Analysis of tagging data has shown that 11% of Pacific halibut are recovered in different IPHC regulatory areas than the ones that they're released in. And we also know from some recent larval induction modeling work that there's large amounts of larval exchange between the Gulf of Alaska and the Bering Sea. And so these fish are moving around. There's plenty of opportunities for gene flow across all life stages. And then, taken in, management or assessment context, these results are consistent with current assessment practices that, model the stock as a single coast-wide entity. So, yes.
So is that it?
What?
End of story. Done.
Well,
No stock structure.
yeah.
How does it continue?
I mean, that's a good question. I think it is sort of sticky, because by failing to detect something, doesn't necessarily mean you've proven it to be a fact, right? So
There wasn't isolation by distance, which one would expect based on.
yeah, I think so we've done some of those analyses, but we need to, I guess, spend a little more time. But there's
I think actually might be interesting stuff to publish. Because people talk so much about population substructuring.
Yeah.
And
Right.
Also the opposite. The opposite is also
yeah. No,
Over thousands of kilometers, right?
yeah. Certainly, no, definitely. It is kind of an interesting scenario because that is something that is lacking.
similar to what, at least the work that's been done so far at Sablefish, for example, too.
Right.
Well,
I was gonna say, yeah.
paper on stable
Is
fish.
that you?
Yeah. That was,
Set yourself up here.
right. So yeah, I'm very familiar with that. Yeah, that was my master's thesis, and there was a paper that actually was just recently published using the same techniques we're using here that corroborated that. So so yeah, it's not. Yeah, where we go next.
Find a new problem.
Right.
What's the minimum number of individuals per generation moving that would result in this?
Well,
It's pretty small.
it can be very small, but it's hard to say, like, because in genetic terms, like, it's a function of like effective population size and migration rate, right? But like effective population size is often very disconnected from like sensitive population size or even number of breeders. So yeah, to just say, here's a number of individuals, it's more of like a function of migration rate.
So this still doesn't disprove the possibility that they're demographically isolated.
Right.
I'm not saying it's likely, but I mean, this doesn't rule that out.
No, yeah, and I think you have to think here like these sorts of pattern, like in terms, like we're thinking like evolutionary timescales here, which are kind of very different from like annual sorts of like annual kind of management cycle kind of thing, right? So yeah, you can still have very, relatively, I guess, small rates of movement that might be relevant in a management context, but still have, kind of genetic homogeneity existing. This is what I'll say for that.
That's still the case for these analyses.
Yeah.
But it is a really important result that we don't have evidence of unique genetic components that we need to somehow conserve.
Right.
There appears to be a pretty broad genetic pool, so we don't.
Which is quite relevant to what we were talking about before.
It is.
That
We still may want to
structure.
conserve spatial structure to produce recruitment, because they can be the same fish in the right place at the right time. But we don't. It's not unlike salmon. We don't have evidence where we have to keep fish in this stream because they're genetically different than the one next door.
Right. We want to keep fish in the stream because they might hit it big next year
Exactly.
under a different environmental regime.
And there's also ecosystem considerations, like you don't throw off the balance of where health tend to be. Does this have any implications for post-skin market capture?
Not necessarily. I mean, as far as like, as far as my understanding goes, I mean, I think it would probably simplify things because you don't necessarily have to account for stock structure, right?
Matching recruits to particular populations.
Right.
But this doesn't mean that CKNR is not going to.
No. I mean, yeah, that's, you're estimating, like kinship relationships, right? Rather than trying to look for, large sorts of discrete population groupings. It's, I guess,
So I think this probably makes it easier.
in my head, they're more of like, kind of like forensic sort of techniques, right? You're looking at relationships between individuals, between, large patterns in the data set.
So you have plans to publish this anytime soon?
Yeah. Oh, yeah. Be ashamed to go, to not. do that.
Definitely will.
Then yeah.
Or do you want to talk about next?
Oh, where to go next? Yeah, I mean, that's a good question. I mean, another, thing to do, we do have some data from juvenile fish that might be kind of another interesting avenue because that's, another way to potentially like look at stock structure before those fish, it's like they haven't had as much time to kind of move around and mix, right? Like if for some reason, say we're, missing it in this data set. But yeah, so I think that could be, kind of another piece to possibly support this conclusion. But yeah, as far as, where to go next, yeah, certainly open to discussion there.
Yeah, in addition to that, we're also considering looking at the population extremes. So comparing, genetically speaking, samples from the northern edge of the population, right? North of 60 degrees north,
Yeah,
and states that were caught in the most southern edge of the distribution to see if there's any age effects, differential, anything that we can actually see.
right.
It's different. I didn't notice in the details of what you were showing, but I remember, might have even been last meeting, we were talking about 4B. Possibility that that's a different stock. Is there
Yeah.
any indication of that?
No, not here. So, I mean, yeah, I guess the whole premise there is so the kind of most recent. So in 2016, there was a genetic study published on halibut and found some evidence of genetic differences in that area. That study used samples that were collected in the summertime in that area, and the rest of the data, like we're actually using some of the samples that were used for that study here. So this was put together to take another look at that using your genomic techniques, basically. But there's really no difference between the samples collected east and west of the Anchica Pass, like in this analysis here. So, revisiting that with newer techniques, higher resolution techniques, and samples collected during the spawning season in those areas. This analysis doesn't support that.
There you go, Ian. Makes your life easier.
Russians are fishing on the same stock.
Well, I think that's a much harder question to answer because, we don't have samples from over there, right? So,
Get samples pretty close to it.
well, pretty close, but, the distribution extends quite a ways, past where sampled. So
How close to northern Japan is the distribution?
I believe it pretty much extends to down south to Hokkaido.
Yeah,
They get
north of
samples for
Hokkaido.
you?
Yeah, if you, yeah.
Do that next week.
Some,
Need you to be there in the winter.
chromatography. Okay. Thanks, Andy. So yeah, next is the research area on reproduction, and we've discussed that quite a bit in the past, particularly with regards to all the work that we've done in probably the last 5-7 years in setting up all the background information on the reproductive biology of species to be able to use histological based analysis to refine our maturity estimates. So this is really what we've been focusing on, is really the revision of the maturity schedules based on histological assessment. And that's what Colin Jones is going to talk about.
Yeah, so I guess I'll just start off by saying that, we presented a lot of biological data in the document 09, but for the presentation purposes, we're just going to be focusing on the modeling aspect of the maturity data, because that's going to be what's directly inputted into the stock assessment. So I think that's where we're looking for the SRB to provide most input into what type of model should we be using to create our ogives. And then also, I'm going to be discussing how we're creating our coast-wide ogive from the regional curves. And we'd like some input from the SRB on that as well. So just to provide some quick background on where we've been and where we are now. So previously, over decades, we've been taking visual field estimates from our fishery independent setline survey to classify our current maturity estimates for females in Pacific halibut. And so currently we have 4 different categories for female reproductive development using visual estimates. And that's in these pictures here. So the first one on the left is what we classify as immature. And then the 3 on the right would be mature individuals. And then that is what is going into our current maturity estimates in the stock assessment. So just as a quick reminder, so this maturity ogive that I show here on the right was taken from data collected in 2002 and 2003 from the survey, from visual estimates off the survey. And so this maturity estimate hasn't been updated in over 20 years. And so to use more modern techniques, what the IPHC Secretariat started doing was looking at histology. And so starting in 2017, we went out and conducted a reproductive assessment project that allowed us to classify the reproductive, basically the female reproductive cycle using histological methods. And we have basically 9 stages here. The one on the top is primary growth, which goes from down to the cortical alveolar stage, which histology-wise we classify as immature females. And then as you get to secondary growth, which is the beginning of vitellogenesis and moving through oocyte maturation, we classify the females as mature. And so it's also note that females are assigned to a developmental stage based on the most advanced oocyte stage present. So you could have a couple of different stages present under a microscope, and we classify the female as what's present in the highest developmental stage present on histologically. So what we did was we set out to collect coast-wide samples starting in 2022 on the Fishery Independent Set Line Survey, collecting ovary sample collection for histological assessment. And we had a really, I would say, pretty successful sampling for that year. So this was basically the last FIS that we've had that we were able to sample across all 4 IPHC biological regions. And we were able to collect over 1,000 histology samples. And those red dots indicate a distinct FIS station where a sample was collected. And I believe it was 489 different FIS stations got a histology sample in 2022. And as you can see, we got over 400 in biological region 2, 351 in biological region 3, 181 in biological region 4, and 51 in biological region 4B. And that was, and 4B was actually a 1-to-1 sampling rate. So they only caught 51 females. It's because the FIS, if you know, tends to catch a lot more males on 4B than they do females. So here we're showing the raw data from the histological maturity assessment by biological region. So as on the top is biological regions 2, 3, and then on the bottom is 4 and 4B. And each bubble represents a specific age. And so this is just to kind of give you an idea of the distribution of the data by biological region before fitting a model to it. And as you can see, biological region 2 almost looks like kind of like a linear line going up, towards from about age 8 to age 20. Biological region 3 definitely has a steeper, more earlier development and maturity compared to biological region 3, biological region 2. And then region 4, and then 4B, the sample size is small in 4B, just to know. But this kind of just gives you a kind of a general kind of first look at what the data looks like before model fitting. So what we did was we looked at a few different types of model comparisons. What we're going to, the first 3 dots on the top there, the logistic GLMs, looking at age by region as well as log age by region, and then the logistic GAM with a smooth function, looking at log age by region. Those are the ones we're going to focus on today in the presentation. We have briefly looked at a couple of different types of breakpoint models, one with an estimation, and then also looking at a breakpoint at the age of 50 maturity by region. So we can get a little bit more into that at the end, but we're not going to present the actual modeling of that today. So to start, we're going to look at the the logistic GLMs. And this table on the right was something that we presented in document 09. And this just kind of is a good reminder of, we looked at age, length, and weight and their fit to the model. And this shows that age is a better predictor of the model than length and weight. And so we're going to focus on age moving forward. We did provide some of the figures for length and weight in that document. Ian was happy to see this because age is what we're currently using as an estimator in our model for the assessment. See here. So this was the first output. This was the GLM, just looking at age. So no transformation of the age data by region. And yeah, we have, I'm showing the AIC values here just as a reference. So 894.2. Also, it's important to note that the bubbles here are the bin data. So each bubble has at least 10 fish, except I believe the last bubble, which is usually on the top there. So towards the older age fish. So the model is actually being run by the raw data, but the data is being binned for plotting purposes, just so you're aware of that. And so you can see here, looking at age by region, with no transformation of age, the model does not fit very well towards the very young ages. Oh, and also I forgot to mention those dashed lines. So those dashed lines, there's 3 of them. And those are showing age at maturity at 5%, 50%, and 95%. So it kind of gives you an idea of the distribution of the data by region. And so you can see that there's a pretty wide distribution in region 2, from about age, just under age 5 to age 23 to A95. And then as you move down to about region 4B, it's a very tighter fit, which means that the steepness of the curve is a lot more steeper. But then as we move towards the log transformation of the age, you can see that the logistic GLM does a lot better at fitting the model towards those younger ages. And so it also improved the AIC value by about 20 points. And so that's why we're going to be using log transformation of age moving forward, because it definitely fits the model better, as you can see here. And then using those regional curves, what we did was we overlaid them. So this is those same 4 regional curves with the confidence intervals overlaid on top of each other, so you can kind of see the different distributions. And so using this, what we ultimately wanted to do was create a coast-wide ogive, because that is what ultimately is going into the stock assessment. And so in order to do that, we calculated it from weighted regional curves using the Fisher Independent Setline Survey space-time model, numbers per unit effort, or MPUE. And the reason why we're using MPUE and not WPUE is because currently in the stock assessment, the total reproductive output is estimated by multiplying the number of reproductive fish at each age bin by their average somatic weight, and then by the fecundity per age or body weight, which is currently estimated at 1. So defining our coast-wide ogive in terms of number of fish is consistent with what is currently being used in the stock assessment. And so as you can see here, the coast-wide ogive is roughly falling in between biological regions 2 and 3, as you would expect, because that's where the largest numbers of estimated biomass, estimated numbers of fish is coast-wide for Pacific halibut.
But that's not necessarily, like, why use NPU? NPUE is an index of density, which is not necessarily a good weighting factor, because you could have a very dense population in this tiny area. So you would
We're
you
not
want to weight
using NPUE, we're using the numbers estimated for each region.
the total
Right.
numbers.
Total
Number
number
of fish
estimate.
estimated for each region.
Okay, so not NPUE.
Yeah, well, I mean, it's based on
It's
NPUE.
based on the, yeah.
Right. So it is actually the numbers.
Yeah.
Because you're right.
It's-
Otherwise it would be.
Yeah. It's numbers of estimated numbers of fish per biological
And
region.
the weighting makes sense.
Yeah.
And those numbers are above, is there a threshold size for those numbers, or even small reproductive fish count those numbers?
Yeah, let me think about that. Yeah, I guess that's not being factored in here, is
We're
it?
using everything we catch on the survey. The curves are based on what we catch in the survey. So how you would filter out, because we're using immature fish and mature fish to fit the curves.
Right, but then you're weighting the curves for the,
Yeah, you could get a lower NPV depending on your selectivity on that, right? Selectivity. You have one coastwide selectivity curve. That's
We don't know we assume that the selectivity is the same across regions and that's the availability that differs by region, but we don't really have any way to quantify that. There was a little bit of work that Ray did as part of the tagging study, showed that there could be differences in selectivity, but it's hard to say. Gear is the same in all the areas, so presumably the contact direct selectivity would be the same. There's definitely differences in spatial availability. I don't know what you would use as an alternative to weighting other than what we caught in the survey. Yeah, because it's got to include the whole population range.
Yeah, so I will say that, a lot of, when creating regional maturity ogives or coastwide ogives, like if there was a fish on the west coast of the US, a lot of times the data has just been pooled to create that curve. And so we have the ability to, because we're not sampling at an equal rate across the regions, we're able to use our space-time model index to create that coastwide curve. So I think it's a pretty valuable resource for what we're being able to use it for here, compared to a lot of other maturity ogives that have been created for other fish species.
yeah, that maturity varies regionally. We can create a weighted average.
Yeah. Yeah. And so using the log age by region GLM, the age at 50% maturity was estimated to be 11.3. And so our current estimate right now on the stock assessment is 11.6. Now, just to note that this is only 1 year's worth of data, so it's something to keep in mind. Now, our current estimate seems to be similar, but also it's the total curve that matters. It's not just the midpoint, right? So,
Is the maturity across regions varying in a predictable way? I mean, is it areas with warmer temperatures maturing earlier?
that's definitely something we could look at. We have water profiler data from the couple of years. Unfortunately, we're not collecting water profiler data this year on the FIS.
Well, we know whether
But-
regions.
But yeah, it's definitely something we could use potentially as a covariate or something like that to look and see whether or not we can better predict maturity through environmental factors and those kinds of things. But that's just not something we've gotten to yet.
Just at a glance, they don't line up 2, 3, 4, 4B.
No, definitely not. Like it shows here, there's definitely currently we're definitely seeing earlier, younger maturing fish in region 3 compared to region 2, which definitely could have some implications, especially if we keep seeing that trend over time. So it'll be neat to see as we get more years worth of data whether or not these trends hold.
And there are also 2 parameter models, so it's not as simple as maturity earlier.
Yeah.
Look at that comparison between 4B and 4. They start maturing earlier in 4. They
But then
reach it, 100% maturity at a later age.
at a later age, yeah. So then the next thing we wanted to look at was a GAM. So this was using the MGCV library and the GAM function in R as a smooth function with log Asian by region. And so this gave an AIC value, a little bit better fitting model than the GLM. And then we set the K or the basis value at 14 to allow the model to enough smoothing or what people maybe call wiggliness of a GAM without potentially overfitting the model. Because sometimes you can have that K value potentially too low, and we actually did run into that with particularly in region 4B. If you drop that K value a little too low, it actually started showing weird, not biologically relevant because it was overfitting the model. And so here you can see that the GAM overall does a better job at the very younger age, maturity at young age of female Pacific halibut. But one thing, the most important thing out of this is biological region 3. You can see that, so the GAM does an estimated degrees or effective degrees of freedom by region, and which is basically like the wiggliness of the curve by the region. And so if you're close to a 1, that's basically a linear model. In biological region 2 was like 1.03. So the fit of the GLM and the GAM was almost practically identical in region 2. But in region 3, the effective degrees of freedom was just over 5. So it definitely, I don't know if I have the cursor here. You can see here that there's definitely a lot more wiggliness here in the center of the curve, close to where that 850 value would be. And then also as you move older fish near the top there, you can see that there was definitely a dip in the asymptote of the curve there as you get towards older fish.
Yeah, what's going on there? Is that evidence for skip spawning or?
There's potential. So we just had a little discussion about this last week. So I think what's ultimately happening here is that you see that there's a large cluster of individuals right here. So it's reaching 90% maturity pretty fast in region 3, at least from the 2022 data. But then there was a couple of, I believe, 20-year-olds that were immature. And so that's what's dropping that circle there, kind of sitting by itself.
And in region 2 and in region 4. That's pretty consistent.
Yeah. So it could be two things. One is potential for skip spawning. Another is also the temporal variation of sampling within a year and when the sample was collected. So you could have individuals that you could be collecting in mid or late July that are showing immaturity, but still have the ability to reach maturity by the spawning season. And so that's definitely a confounding, and that's just, I mean, it's just part of that's.
Well, I mean, you could filter, right? You could only fit this
Yeah.
data to ones from later in the summer where if they're not mature then they're not spawning.
Yeah, and that's something we have discussed about potentially looking at. Now there also has been, like Alan was on the Pacific Hake assessment team this year looking at, and they've just recently started incorporating like calendar day into their model output, and basically looking at what point do they hit, would they assume that it's like 100% maturity, and then past that day, you're just assuming that all individuals are mature. We just don't have enough years worth of data in order to do that. That's something that we're going to probably be looking at once we get more years worth of data, looking at calendar day potentially.
So on region 3, would you call that biologically plausible?
I definitely think that is plausible. I don't see any reason why it's not.
If you invoke skip spawning,
Yeah.
evolutionarily it wouldn't make any sense for fish to never mature. But at a population level, it seems possible that you have not 100% of the fish mature every year or spawning every year.
So I'm thinking about the notch or the, whatever it is, that little hump that the cursor is currently on at 850. And then the dip up there.
I think an asymptote less than 1 is plausible. I think the dip is just the chasing.
Yeah. That's what, yeah.
Yeah.
Yeah, unfortunately, the sample size after age 18 is quite low.
Yeah.
Yeah.
And as Colin mentioned, that could be driven by females that are actually slower maturing, not necessarily skip spawning, slower maturing.
That haven't matured by age 20.
Yep. We do have
At the
we
time-
do.
At the time of collection.
Saw this in some Greenland halibut as well. So, not even close to
Yeah,
100%.
Yeah, there's one or two one or two specific individuals that are over the age of 20 that have no sign of development. Now that could be a potential senescence thing, or they just never matured.
Collected during a time of year when they should have.
There's a couple of individuals that are in their 20s that I don't think they've never become mature females. Yeah. So it's plausible. Biologically, it happens.
I'm sure it could happen. It seems like a really bad strategy. That should be
Yes.
strongly
Slow it
linked
down
again.
pretty fast,
Yeah,
right?
Something they ran into something that caused them to not.
Period, and, oh,
Yeah. So it's definitely
or it could be one of those
Biologically.
ones that's gonna grow 7 feet long and mature when it's 30. Right?
Yeah. And so then we wanted to show the difference between the two model outputs, so you can kind of see them overlapping. So this is the log age by region, GLM, as well as the GAM. And as you can see, that definitely towards the tail ends of both of the curves is where the GAM is different. The GAM does a better job of showing that initial rise at younger age females. Because currently, right now, in the stock assessment, the ogive is assuming that no females under the age of 8 are mature. It basically goes like straight up like that. Now that holds with the 2022 data, but I've been starting to go through the 2023 samples, and we have been finding 7-year-olds that are mature. So it's definitely there's definitely that data is that biologically they are maturing at a little bit younger age, I think, than what we originally were thinking.
I guess I'm not concerned about the right-hand side here because that's multiplied by a very small number of individuals, but the left-hand side, that could matter, right?
Yeah,
because
yes.
there's a lot of fish in that stage.
Yes. Yeah. Yeah.
Which is exactly why we started to explore the GAM, because we were worried about the lack of fit to the 5, 6, 7, and 8. With just assuming the 2-parameter logistic.
I mean, the 2-parameter logistic is constrained to be symmetric there.
Exactly.
So yeah.
It's going to be
Biological
driven by
reasons.
wherever the data are, as opposed to
Except we've taken logs, so that.
better. But we recognize it's only 1 year of data too. So
Yeah.
But it is, I mean, that is a common problem. Seeing lots of maturity data. They're not symmetric.
Okay. So then, just to
Go back on those two. Did you run the stock assessment with both of these to see?
no, that'll be something that we'd be doing, hopefully including the 2023 data, but at some point, that's what we'll be looking at this fall. These are hot off the press.
Yeah. Yeah, literally in the last couple of weeks. Yeah, so just to continue on here, I got to think just a couple more slides. So we once again set out to collect more histology samples on the FIS in 2023. Unfortunately, last year was a reduced design only in biological regions 2 and 3. So as you can see here, 2023 is designated by the blue dots. So you can see the distribution of samples. So if you combine 2022 and 2023, there's definitely really good geographical coverage in those two regions over both those years. We were able to collect just over 1,100 samples last year on the FIS, 400 in biological region 2, and then just over 700 biological region 3. So there's definitely a good data set to work with for those two biological regions. Unfortunately, we're suffering a little bit on trying to get samples out west. But then for this year's FIS, where sampling is going to be occurring or currently occurring, because boats are on the water, is where those green circles are. And our targets are 400 for biological region 2, 400 for biological region 3. And then we're hoping to get 550 out in the Bering Sea. Currently, they're going to have a sampling rate of 1-to-1. So every female that catch, they're taking a sample because we just don't know when the FIS might go back out there. So we're going to try to get as many samples as we can. And then, yeah, so just in conclusion, as you saw, that definitely the transformation of the log gauge definitely helped fit the model. And I guess where we're looking for the SRB is some kind of direct input on your thoughts on the GLM or the GAM or any other suggestions of modeling that we could do this summer. And then also if you potentially are endorsing the way that we're formulating our coastwide ogive or any other ways that we might be able to model that better. Because the 2023 data is currently being processed and will be completed this summer. So we would like to hopefully present 2 years worth of data to you guys in September. And if we had a better idea of what type of model we could use for both of those years worth of data would be helpful in our outputs for this summer for you guys in September. So, yeah.
You'd be interested in doing it on adding 2 more models to the ensemble, would you?
Well, you could certainly go that route. I mean, there's several considerations before we get to sort of production line use in the assessment. The first one I think that we're focused on is how do we characterize what our best understanding of maturity schedule right now? And then we need to think about, well, how would we link that to the historical period? Would we just replace the old curve, or would we, do we make the assumption that nothing's changed and nothing is changing over time? And that's where we wanted to see at least 1 more year of data to understand the interannual variability. And then we need to think about how this would potentially, whether we would need to monitor going forward, collecting more data or at some period of time. But we really need to know what the interannual variability looks like. Because in the assessment, it's easy for me to model it as a year-specific maturity curve, if that's a meaningful thing. But we obviously don't want to just be fitting to noise. With 1 year of data, every age is confounded with a single cohort. So we don't necessarily, it's not necessarily the best characterization of a global maturity schedule, at least until we get another year's of data.
What's the original curve based on? How many years of data was that?
2 is 2002 and 2003. Just from regulatory areas 2C and 3A.
But it was consistent with,
It
they
was.
did a qualitative
It was,
comparison
yeah.
across a bunch of years and a bunch of areas. And the maturity at age has been incredibly conserved for this species. Despite huge changes in size at age, maturity at age has hardly changed at all. And it's a little suspicious why we wanted to go the histological route to make sure we weren't somehow characterizing it wrong. But at least at first blush, it seems like we're in the ballpark.
Both
conserved, but also variable by region, but not because of differences in growth rate, which is, I think,
Yeah.
really interesting.
Yeah.
Right? I mean, regional effects that are
Right.
explained by differences in growth rate, sure. Then you just use
Now,
rate
remember,
rather
these
than
fish
age, but that's not
move,
what's
so
happening.
some of the
Yeah.
differences by region could be that fish that are on a fast growth trajectory and maturing early or late may end up in some areas rather than others.
Yeah, and that's why with multiple years, you estimate the cohort effect. Right.
So, I mean, I think at some point here in the next few months, we'll try this curve out in the assessment and see how much change it makes. My guess is it's going to mostly just be at the scaling. It's going to get scaled away. Where it really gets interesting in the assessment is if we start to see trends over time, or something that we really believe to be meaningful differences between years and cohorts.
Yeah.
Then it starts to make a big difference.
Especially if it's linked to
Yeah.
size.
I mean, looking at those differences, you're talking about a difference of 0% or 10% mature at age 7, 8, something like that. Those are small fish anyway, right? They're abundant. But
But they don't have
But
a
they're-
huge egg out.
exactly.
And we have a very weak spawner recruit relationship to begin with, although it could, if we're characterizing this wrong, that might actually get better.
Well, that's another thing we're hoping to look at. The fecundity
Fecundity is the other link. This is going to be potentially important.
is you could look and see whether or not age has a huge factor on egg output.
And I did a couple of years ago, I did a sensitivity where I added a fecundity relationship. I said, what if older females were more fecund per pound body weight? And I looked to see, could we fit the data better? Would we reduce the recruitment variability by explaining, basically getting the x-axis of the spawner relationship more correct? And I didn't find any evidence for that, but that's a very weak test, right? It's just
Estimating that cohort effect is going to require a lot of data. It's not just adding a couple of years.
No, it's a long time series. Yeah.
right, but at least with 2 or 3 years of data, we're not completely confounded between age.
You're not gonna
Some of the variability on those curves could be real variability among cohorts. If there's a more abundant effect, right now we have the 2012 year class is relatively abundant in the data. So in this information that would have been 10 years old, cohorts on either side of it, particularly on the older side for a couple of years, are much smaller. So it's conceivable there could be a density dependent cohort effect there. We won't really know until we see information. Like you said, we won't really know about cohort effects. Go off the data.
I mean, this is really, really cool, and I think really directly linked to this assessment.
The quantitative science guys have been ecstatic about this new data set and
Well, also relieved. Yeah, we weren't
Yeah.
sure what we were going to find. It's
yeah,
really
yeah, we could have
5
got
years
something
off.
that was just like, yeah.
You don't have to deliver the, oh, by the way, the SSB estimate is.
Not
I've been warning people for several years that if it's off, this is potentially pretty important to our understanding of reference points and stock status.
Not an exceptional circumstance.
No, not yet.
So this is not going in the next assessment. Is that right?
Well, it could, but we were reluctant to revise our whole perception of maturity based on 1 year data. We want to see at least a second.
This doesn't revise a lot.
Well, I mean, we haven't seen
Not
the
Really.
existing maturity of the job that's in assessment.
That's the one I showed at the
That's the
beginning.
average.
If you go back
You
to
don't
the,
have the overlaying.
no, I don't.
We made that one. It's
Oh,
not.
I
all that
did.
different.
He did make
So
that
the same,
one.
initially it's the same.
The center is almost exactly the same. It was 11.3.
I could do that.
versus
I could do that really
It's
fast.
dropped
actually.
a little bit, but it's on the direction of more younger fish mature and fewer older fish. So if anything, it's I think going to make things a little more.
I wonder if the model would improve if there would be interaction of age and length.
Did you run some interactions between age and length, Ray?
We did include length in the model as well. It complicated the story, but adding length to the model.
It proved a fit.
Improved the fit.
Yeah.
So that it seemed to be I think it was to be a fish, sorry, a larger fish, more likely to be mature than a small fish for a given age.
Just normal.
And the model would come up with 300, no reserves.
Yes.
Yeah,
Which is gonna make your assessment more complicated.
Well, and the weighting now becomes complicated as well. Think about trying to weight that. Yeah, that's starting to get a little circular though, because then we apply it back to numbers and assessment.
Biomass is actually not. So,
Yeah.
How are we doing with time?
we're done. I think we're
For today,
done for today. We have to get started on the ground. Done today?
but we should still be ahead of schedule because
Yeah.
we have a whole period tomorrow afternoon from 1:30 to 4 that's open. And we're, if anything, we're ahead on our the other sections because Basha's already given her presentation.
And where are we on the biology and ecology? Percentage wise, how far along are we?
We probably have maybe 30% left. That's,
Percent left?
Should be fine tomorrow.
probably about 30 minutes with discussion, 45 minutes with discussion.
That's where we're picking it up tomorrow on mortality.
Release mortality, fishing technology.
Sounds good. And we're meeting at 7. What time is dinner?
Yeah.
9 o'clock start again tomorrow.
9 o'clock start tomorrow.
9 o'clock tomorrow.
Thanks, everyone.